Hello students, this is Dr. Dakshayini, Assistant Professor from Department of Zoology, Maharani Science College for Women, Bengaluru. Students, in this practical class, let us study the different types of poriferans. The contents as follows: Introduction to phylum Periphera and its classification, study of different types of poriferans. Phylum Periphera comprises the lowest multicellular organisms and it belongs to kingdom Animalia. The term Periphera was coined by Robert E. Grant and it is derived from two Latin words. Porus means pore, fera means to bear. Therefore, the term Periphera means pore bearers, and their body possesses numerous surface openings. The poriferans may be defined as an asymmetrical or radially symmetrical multicellular organisms with a cellular grade of body organization without well-defined tissues and organs, which are exclusively aquatic and sedentary forms, and they are solitary or colonial organisms with body perforated by pores, canals and chambers through which the water flows, with one or more internal cavities which are lined with coinocytes and with a characteristic endoskeleton made up of calcareous spicules, siliceous spicules or horny fibers of spongin. The characteristic features of phylum Periphera. Poriferans are exclusively aquatic forms where majority of them are found living in marine waters except one family called Spongelidae which lives in fresh water. They are sessile and sedentary forms and they grow like plants. The body shape is vase-like or cylinder-like, shows asymmetrical or radially symmetrical body structure. The body surface is perforated by numerous pores called ostia through which the water enters into the body, and one or more large openings called oscula by which the water exits from the body. The multicellular organisms with cellular grade of body organization has no distinct tissues or organs. They consist of outer ectoderm and inner endoderm with an intermediate layer of mesenchyme. Therefore, sponges are called as diploblastic animals. The interior space of the body is either hollow or permeated by numerous canals which are lined with coinocytes. The interior space of the sponge body is called spongocele. One of the interesting characteristic features of sponges is that they possess endoskeleton which is made up of the flexible spongin fibers, siliceous spicules or the calcareous spicules. In sponges, the mouth is absent and the digestive system is also absent, where the digestion of food takes place inside the cells called the collared flagellated coinocytes cells. Hence, the digestion is intracellular. The excretory as well as the reproductive organs are also absent in sponges, where these physiological process takes place through a simple diffusion method through the body surface. The nervous system is also absent in sponges, where the nervous and sensory cells are probably not differentiated, if at all it is present. All sponges are monoecious in nature, means they are hermaphrodite. The reproduction takes place by means of sexual as well as asexual methods. Asexual reproduction takes place by forming budding or the buds, and also by gemule formation. And the sexual reproduction takes place by the production of ova and sperms, where the fertilization takes place internally and it is cross fertilization, and development is said to be indirect, where it passes through the free swimming larval stages. And in sponges it shows two types of larvae, the Amphiblastola larva and the Parenchyma larva. The sponges possess a high power of regeneration. The organization of sponges are grouped into three types, which are Ascon type, Cycon type and Leuconid type, based on the complexity of the infolding of the body wall of sponges. And few examples of phylum Periphera are Cycon, Grantia, Euplectella, Hyaluronima, Spongela, Euspongia, etc. Coming to the classification of phylum Periphera. This phylum includes for about 5,000 species of sponges, which are grouped into three classes, mainly depending upon the type of skeleton found in them. The three classes are class Calcarea, class Hexactinellida and class Demospongia. Class Calcarea. The members of this class are exclusively marine forms. They have a small sized body and they are calcareous sponges measuring about 10 centimeters in length. They are solitary or colonial, having body shape either vase-like or cylindrical. They show asconoid, cyconoid or leuconid type of canal system. In these sponges, a skeleton of separate 1 or 3 or 4 rayed calcareous spicules are present. Example, Cycon, Grantia. Class Hexactinellida. The members of this class have moderate sized body and some may reach for about 1 meter in length. They are found living in deep tropical seas and they are commonly called glass roe sponges. Their body shape is either round or oval-like and their skeleton consists of Siliceous spicules, which are trioxan with 6 rays. In some, the spicules are fused to form a lattice-like skeleton. And there is no epidermal epithelium present in these members. And the coinocytes line the finger-shaped chambers. And members of this class shows lacunar type of water canal system. Example, Euplectella and Hyaluronima. Class Demospongia. The members of this class generally marine forms and only few are freshwater forms. And this class contains the largest number of sponge species. And the members of this class body size varies from small to large. They may exist either solitary or in colonial. The body shape may be vase-like, cup or cushion type. And the endoskeleton consisting of the siliceous spicules or the spongin fibers. And they show lacunar type of water canal system. And the examples are Spongela and Euspongia. Students, let us study the different forms of sponges from each class of phylum Periphera. The Cycon, the systematic position of Cycon is, it belongs to phylum Periphera, class Calcarea, order Heterocela and genus Cycon. Now it is called Scypha. Scypha exhibits the first stage of body wall folding and is therefore called cyconoid sponge type. Scypha is commonly known as urn sponge due to its urn shape, which resembles the water vessel, and it is also called a crown sponge as the fringe of long and straight spicules looks like a little crown at the distal end of the body. Scypha is a marine sessile sponge and it remains permanently attached to the submerged rocks or other solid substratum. It is widely distributed and is especially found abundant in places where the wave action provides adequate amount of food supply and with well oxygenated water. It is a branched colonial sponge. Sometimes it also exists in solitary form. Scypha has the structure of solitary or branched cylinders, each measuring about 2 to 7 centimeters in length and 0.5 to 0.6 centimeters in diameter. Each resembles a slender vase-like cylinder or slightly bulging in the middle. All the branches are connected together at the base, which remains attached to the substratum, where one or more buds may be found on the sides of the body. The cylinders are flexible and they tolerate the high wave tidal. This Scypha color ranges from grey to light brown depending upon the species and its surroundings. The surface of the body is perforated by numerous pores called ostia, and the distal or free end of each cylinder has a single large opening called osculum, which is surrounded by a oscular fringe of monoaxon spicules. The body is supported by calcareous spicules, which is consisting of monoaxon, triaxon and tetraxon type. It has a large central cavity called spongocele, and Skypha shows Cycon type of canal system. It reproduces both sexually as well as asexually. Sexually, it reproduces by means of budding and forming gemmules during unfavorable conditions. The sexual reproduction takes place by means of producing the gametes. The fertilization takes place internally, and development is said to be indirect, where it passes through the amphiblastula larva. The Skypha shows high power of regeneration. Transverse section of a Cycon. The transverse section of a Cycon shows the following histological details, where the section is circular in outline. It shows the body wall, which is in diploblastic in condition, composed of outer dermis, which is made up of flat cells called pinacocytes, and it consists of inner endoderm, which is made up of a large flagellated collared coinocytes. It also lines the spongocele. Between these two layers, there is a mesenchyme which contains calcareous spicules and amoeboid cells and many other cells such as trophocytes, thymocytes, coloncytes and archaeocytes. The infolding of body wall gives rise to the incurrent canals and radial canals in Cycon, where the incurrent canals are lined by pinacocytes, and these canals are exposed to outside environment. The radial canals are lined with coinocytes, and it is exposed to interior of the body space called spongocele, and the openings of radial canals into spongocele is called apopiles. The radial canals alternate with incurrent canals, which are communicated by prosopiles. The radial canal opens into the spongocele by internal ostium or the apopiles. There is a large cavity called spongocele, which is present at the center of the section, and it shows the cyconoid type of canal system, and the course of water takes place from exterior into incurrent canals through ostia, and from incurrent canals to radial canals through prosopiles, and from radial canal to spongocele through the apopiles, and from spongocele to exterior, it is through the osculum. The Hyaluronima. The systematic position of Hyaluronima is phylum Periphera. It belongs to class Hexaphora, order Amphidiscophora. It is a marine deep sea sponge and it is commonly called as glass rope sponge. And it is one of the commonest hexactylanidan sponges which is found distributed worldwide. The body shows variability in form but usually rounded or oval and radially symmetrical. And it superficially looks like a ball of glass wool with projecting tuft of glassy spicules. The body is perforated with numerous ostia and an apical large osculum. And the endoskeleton consists of some small branching and 5-rayed spicules extending from all over the body, and it, besides having 6-rayed spicules, it also consists of large and small amphidisc spicules. The body is elevated from the substratum by a stalk-like root tuft which is often spirally twisted like a rope, where it is made up of bundle of very long monoactin anchoring spicules, and the root tuft also passes through the body as an axis or the columella and frequently projects above as gastral cone. The middle part of root tuft commonly bears the symbiotic polyps. In Hyaluronima, the canal system is said to be complicated type because the spongocele is usually found absent in Hyaluronima. Euplectella, the systematic position of it is, it belongs to phylum Periphera, class Hexactinellida, order Hexastereophora. Euplectella, it is a solitary marine deep sea sponge and it is commonly called as Venus flower basket, and it is the most beautiful glass sponges. The body is elongated, cylindrical and slightly curved, measuring for about 15 to 30 centimeters in length and for about 2.5 to 5 centimeters in diameter. The skeleton contains the hexactins, that is 6-rayed siliceous spicules. In addition to it, it also contains a lattice work of fused siliceous spicules, where it shows a unique lattice structure of fused siliceous spicules, giving it a glass-like look. And a net of living tissues surrounds these siliceous spicules that is created by the amoeboid cells called arcucytes. Within the tubular net, there are finger-like chambers covered in coinocytes that open into the spongocele, allowing the water to move through the sponges. At the base of the sponge has a tuft of elongated spicules that are fused to attach to the ocean bottom. There is a terminal opening called osculum, and it is closed by a oscular sieve plate which is formed by the fusion of spicules. There are the parietal gaps seen in the meshwork of spicules and are connected with the spongocele. The large spongocele often harbors various commensal crustaceans which feed on the plankton which is brought in with the water currents. The Spongela. The systematic position of Spongela is it belongs to phylum Periphera, class Demospongia, order Monoxenida. Spongela is a common, widely distributed freshwater sponge and it belongs to the family Spongelidae. It is found growing on submerged plants and twigs in ponds, and it shows various shades of green because of symbiotic algae which is present inside the Spongela. Its colony is more profusely branched than any other freshwater sponges, and it has got a characteristic odor. Spongela shows the lacunar type of canal system where the coinocytes are restricted to a small rounded chambers. The endoskeleton consists of siliceous spicules which are made up of monoaxon type, and it also consists of large spicules called megascles, and these are held together by spongin fibers and they provide support for the colony. Spongela is an hermaphrodite where it reproduces both sexually as well as asexually. Asexual reproduction takes place by means of budding as well as gemel forming, and sexual reproduction also takes place. An unusual free-swimming larvae is peculiar to this Spongela. Euspongia. It belongs to phylum Periphera, class Demospongia, order Keratosa. Euspongia is a marine sedentary form found attached to the rocks. It is commonly found in warm shallow waters with a rocky bottom, and body is massive with variable shapes which is often shows globose shape or cup shaped structure, and it is dark brown to blackish in color. The surface of the sponge shows several small projections called the caunuli which are made up of strands of spongin fibers, and body is crisscrossed by many inhalent, exhalent and flagellated chambers and other spaces. The skeleton is made up of network of spongin fibers. This sponge is of a commercial value where the dried skeleton is used as bath sponge and in cushions and in offices for wetting stamps and counting currency notes. And it shows a remarkable power of regeneration where this feature is exploited in spongy culture, and the cut pieces of a bath sponge become large enough to be marketable in about 3 years. As we all know that sponges reproduces both by asexual as well as sexual methods. The asexual reproduction takes place by means of budding as well as forming gemmules. Gemmule is a type of internal bud which is formed in sponges to pass the unfavorable seasons. It is helpful in asexual reproduction, and it is an asexually reproduced mass of cells that is capable of developing into a new organism, that is an adult sponge. Gemmules are resistant to the extreme conditions such as desiccation, freezing, and also anoxia, that is lack of oxygen, and it can lie around for long periods of time. The gemmules are formed in freshwater sponges, example Spongela. During unfavorable conditions, the archaeocytes of the body will group together. These archaeocytes are filled with glycoproteins or lipoproteins. Around this group of archaeocytes, there is a protective layer which is formed, and each gemmule shows a central group of archaeocytes around which an inner and outer chitinous layers. Around this pneumatic coat which is formed, and this coat is supported by a small amphidisc spicules, whereas the outer coat shows an opening called micropyle. When a number of gemmules are formed, the sponge disintegrates and all the gemmules are liberated out, and they lie in dormant conditions on the bottom of the pond. And with the onset of favorable conditions, the gemmules are hatched out and develops into a new sponges. Coming to the skeleton in sponges, the soft gelatinous body wall of sponges is protected by endoskeleton, which is made up of crystalline calcareous glassy spicules, and they are present in the mesenchyme. These spicules are secreted by a specialized mesenchymal cells or the amoeboid cells called scleroblast cells. They may be composed of either calcium carbonate or silica or mixture of both. Accordingly, they are called as calcinated, siliceous or sponginous spicules, where these chemicals are initially deposited around a single thread of axial or axis. Later, the spicule is ensheathed by an envelope. Therefore, the scleroblast exists in two forms, such as builder or founder cell that forms the axis. The thicker cell forms the deposition of helping in the thickening of process. The race of spicules ends in actin or actinals. The spicules are classified depending upon the number of axes or rays they possess. It includes monoaxon, triaxon, tetraaxon, polyaxon, desma, and spears. Since these spicules are larger spicules and constituting the main skeleton of the sponges, and they are called megascles. And the smaller spicules which occur interstitially, and they are called microscleres, that includes spires and asters. The monoaxon spicules. This type of spicules grow in one or two directions along a single axis. The axis may be straight or curved. If it grows towards one direction, then it is called as monoaxon monoactinal. And if it grows in two directions, then it is called as monoaxon diactinal. If the style or tip of the spicule is rounded, then it is called as strongylote. If it is pointed, then it is called oxide. If it is knobbed, then it is called as tylostyle. And if it is thorny, then it is called as acanthostyle. The triaxon spicules. This type of spicules has three axes crossing at right angles to each other, thereby forming 6 rays. The rays may be straight or curved, and they are spiny or knobbed, hence they are called as hexactinal spicules. This type of spicules are characteristics to class Hexactinellida. The tetraaxon spicules. This type of spicules has 4 rays radiating from a common point. If all the rays are equal, then they are called as calthropes. If one ray is long and the other 3 are shorter, then they are called as trienes. The polyaxons. This type of spicules has more number of rays which are radiating from a common point. Desma. It consists of an ordinary minute monoaxon tetraaxon spicules upon which the layers of silica have been deposited irregularly. Later, they unite to form a network resulting in a reticulate skeleton called lithostrid. The spears. This type of spicules have round shape in which growth occurs in a concentric manner around a central point. Coming to the microscleres. These are smaller fleshy spicules which are dispersed in the mesenchyme region. They are often termed as auxiliary spicules as they do not form the supporting framework. They are of 2 types. One is spears and the other one is asters. Spears are spirally twisted, either C or S shaped structures. They are the sigma spires. If they are bow shaped, then it is called as toxas. Or if it is plates with hooks at the end, then it is called as chelas. The asters. These type of spicules have a small centers and long rays, or the larger centers and small rays. If it is oysters, where they are small centered forms with pointed rays. Strongylastus, then they are small centered forms with rounded edges. If it is Tylastus, then they are small centered forms with knob raised. If monoaxons are twisted of a small centered, then they are called as Spirastus. If they are pointed, then it is called as Streptastus. The endoskeleton of sponges also composed of spongin fibers. Spongin is an organic horny elastic substance with scleroproteins, and they are rich in sulfur, iodine and collagens. The fibers of spongins are the thin or fine threads constituting of a soft granular axial core or medulla, which are surrounded by externally by a concentric layers of spongin. The fibers are secreted by flask-shaped mesenchymal cells or the amoeboid cells called Spongiblasts. They later arrange in rows, and the secreted rods of each cell fuses to form a long fiber. They are common in class Demospongia, where the skeleton is of network of spongin fibers. Example, Euspongia. Almost all sponges are provided with a skeleton which is found embedded in mesenchyme, and this may be consisting of separate spicules or of interlacing spongin fibers or of both. The skeleton supports and protects the soft body parts and also serves as the basis of classification of sponges.